Hi everyone, my name is Raina Harris, and I just published a paper today and posted it on the bioRxiv. It actually hasn't been peer-reviewed, so I shouldn't say published, but I made it available for your viewing pleasure. And I've tried as hard as I can to make this reproducible, and so what I thought I would do, though, is make a little video showing you how to navigate the GitHub repository where the data analyses are. So I'll do a screen share so you can take a look. Okay, so what we're looking at now, this is the GitHub repository for the paper. The paper is called Analyses or Analysis of Hippocampal Transcriptomic Responses to Technical and Biological Perturbations. And a link to that paper is in the below. And here we are in my GitHub repository called Disassociation Test. There's a variety of directories where things are stored. This is a little overview of the workflow. So we're looking at RNA sequencing data from 3 separate fields of the hippocampus. And we do 3 different manipulations to these samples. In one, we chemically disassociate the tissue. Another, we expose mice to stressful experience. And another, we put mice through a cognitive training paradigm. And we want to know, is there any overlap in the responses to these that might influence our ability to detect biologically meaningful signatures of cognitive learning, cognitive tasks and learning. The sequencing was done at the University of Texas. We used the Texas Advanced Computing Facility for the computing power. We used FastQC for quality control, CutAdapt for trimming and filtering, and Callisto for counting reads. Then I did everything else in R, data wrangling using these packages. Statistical analyses, I used DESeq2 and a Go MWU package. These are the tools I use to visualize my data, and here's all the places where I'm trying to share it. So basically it's broken down into a few main subfolders. All the data is stored here. The data that's too big is, like the raw reads, are stored in Gene Expression Omnibus, and the Callisto abundance files are stored in another GitHub repository here. The Unix workflow that I used on Stampede is documented here in this directory. And the markdown files are what you can reproduce on your own without needing the Texas Advanced Computing Facility. So let me scroll through this here. What I've tried to do is put each figure on the home page, and you can click here to go view the R markdown file, or you can click here to view the markdown file with all the results and documentation. This is for the effect of chemical dissociation. Then I test the effect of the stressful experience. And then I test the effect of cognitive training. Let me just sort of walk you through these really quickly. Here's one of the R markdown files. This is what you can view to see the code and documentation. The markdown file, the results file, is a little bit more informative and more useful. So here we still have some, I am still describing it a little bit, but you can see results. So I have 9 samples from yoked individuals, 13 from trained, 8 of which came from the C1, C3, and DG. This is how many reads, how many genes we're analyzing, 22 samples total. And this is the Venn diagram output by the Venn diagram package, showing how many genes were differentially expressed by region, by brain region or subfield. Oh, not what I meant to do. And 423 that were expressed differentially expressed by treatment. Here's the hierarchical clustering analysis, and then the bootstrapping to get values of support for the dendrogram. And then a few PCA analyses. Here's one here. Here are the statistics that go with the PCA analysis, if you want to look in detail at that. I ran a lot of statistics on the PCAs. These are some supplementary files. I didn't actually even include them in the manuscript, but if you're interested in looking at the distribution of p-values, these are really helpful. A few more PCA plots because these lower PCAs were informative. And when you put it all together, though, with some help from Adobe, this is the figure, all of those figures above together into one pretty figure. So if you would like to comment or raise any issues, feel free to open an issue or submit a pull request if you would like to change anything. And let me know what you think if you find any of it useful. Right, so that's sort of the GitHub repository in a nutshell. I hope you, yeah, like I said, I hope you find it useful, and let me know if you have any questions or concerns. Thanks.